Hi, and welcome to your biotechnology cloning notes. So these notes are going to go along with the two sheets that you received from me, cloning and cloning continued. So first off, we have to look at what is exactly a clone. So clones and an understanding of clones gets a little misconstrued with the media and Hollywood, but the identical organism from which it's produced. So if you have a turtle and there's another turtle that's produced, it's going to be genetically identical to the original. There are some animals that have been successfully cloned. The first mammal that was successfully cloned was Dolly the sheep. Okay? Dolly the sheep is who I dressed up as for Twin Day during homecoming week. Okay? They have successfully cloned cats, frogs, salamanders, and mice. Okay? You do have to understand the process of how cloning is accomplished. Okay? So it's a 5-step process. Okay? So in your notes there it's 1, 2, 3, 4, 5, and then there's a diagram underneath. We'll look at the diagram on the next slide. So we have to have the nucleus of a body cell. And remember that the other word for a body cell is a somatic cell. So we take the nucleus out of a body cell from the desired organism. And then in a separate organism, we're going to take an egg cell from an adult female. Okay? We will also have to remove the nucleus from that egg cell. So we have a nucleus of a somatic cell, we have an empty egg cell. Those two things are going to be fused together and joined by an electric shock. So we're basically taking the somatic cell nucleus and the empty egg cell, and we're going to place them together and put that nucleus inside the egg cell. After the electric shock, it's going to begin to divide into an embryo. And then that embryo is going to be implanted into a surrogate female or surrogate mother. And that's when the pregnancy actually begins. And that surrogate mother is going to carry that pregnancy to term and will actually give birth to the actual clone. So if we look at the diagram, 1, 2, 3, 4, 5, okay, it goes along with the numbers on the previous slide. So number 1, we have our donor cell, sheep. Okay, so we're taking a somatic cell of sheep 1. Okay, we're going to remove the nucleus of that cell. Sheep 2, we're taking the egg cell from an adult female, okay, removing the nucleus of the egg. Then we take that empty egg and the solo nucleus from sheep 1, we fuse them together. Cell division begins to happen. We get an embryo, and then that embryo is going to get implanted into that surrogate mother or surrogate female sheep. Okay? After the pregnancy has gone to term and the surrogate parent gives birth to the clone, which is the lamb number 5, okay? Number 5 is going to be genetically identical to sheep number 1. It is not going to have any genetic material that is from the donor egg cell, and is not going to have any genetic information from the surrogate mother. Okay? The reason why it is identical to sheep 1 is because we're taking the nucleus from that body cell. The nucleus is where the DNA is stored, and the DNA is your instruction manual for everything else. Okay? Just like with most things, there are people who are going to be on both sides. Either they're going to really want cloning organisms to happen, or they're going to be really against cloning. Okay? A couple of reasons why people and scientists consider cloning is to revive or regenerate animals that are either endangered or have gone extinct. Okay? Reproducing deceased pets. Okay? So if your dog or cat dies, being able to get of that pet. And cloning livestock, drug production, and then cloning within medicine. Okay? There are a lot of misconceptions when it comes to cloning, especially when we're talking about cloning deceased pets or ultimately trying to clone a human. So for example, myself, okay? The clone comes to class with me the next day. There is not going to be another Mrs. Turner. It's going to be a baby, a newborn. Okay? That newborn will have the same genetic information that I have, okay? But we're not the same person. Okay? I grew up in the 90s. that baby, it's impossible for that baby to grow up in the 90s. Environmental conditions and what a person goes through and their life experiences affect who that individual is. So that my clone will have my same genetic material, but ultimately it's a different person. Same thing with a pet. Okay? Your dog, you've had this dog and the dog's been in your family for 10 plus years. The dog dies. You're just heartbroken. You have to have, that dog needs to be with your family. You get that dog cloned. That dog, the clone, is going to be a puppy. That puppy doesn't know who you are, doesn't know any of the tricks or the commands that you've taught it. It is a brand new puppy. The only thing is, it is exact genetically, on the molecular level, exactly the same as the dog, the older dog that passed away in your family. Okay? But a totally different organism that will have different life experiences. Okay? Reasons not to clone organisms. So there is a gamut of reasons of why people are against cloning, or scientists are against cloning. Okay? There actually is a very low success rate. The likelihood of actually getting a clone is very, very slim. It takes a lot of perfect conditions in order to get this to work. Okay? There are moral and ethical issues, the whole concept of playing God, changing genetics that were initially put there and altering them to our liking. Okay? Legal issues, how cloning technologies are to be regulated by the government, or if the government should even be involved. Social issues, how cloning, the new cloning technology is going to affect the society as a whole. Right? All these different things that we have to think about, and we're going to go more in depth with pros and cons of different biotechnologies when we get into bioethics at the end of this particular unit. So at the end of the class period, you got this packet that has a picture of these mice that you see on your screen. Okay, so very similar to the cloning of the sheep diagram that's in your notebook, okay, except for these are mice. There's a color coding page, which is page 1, that goes along with this. Okay? So it's asking you to color different parts of the diagram. Okay? So you have your nucleus donor, you have your egg cell donor. The egg cell from the bottom mouse and the nucleus from the top mouse get fused together with an electric shock. Okay? This embryo gets implanted into the surrogate parent. The idea here is you need to figure out what color mouse 5 is going to be. Okay? Remember where the cells are coming from or what is in the cells to be able to determine what color mouse number 5 or the mouse pup is supposed to be. Okay? Hopefully this helps. Okay? Just again, trying to go over the cloning bit so that everybody's on the same page. Okay? Have a great day, and I'll see you in class tomorrow.